Buenos días y bienvenidos a esta sesión especial de Incliva, en la que contamos como ponente con el doctor Adrià Cañellas, del grupo del doctor Eduard Batlle del IRB de Barcelona, que va a hacer una presentación basada sobre todo en su tesis doctoral, de la que tuve el privilegio de ser miembro de la comisión evaluadora, sobre un modelo de cómo se producen las etapas iniciales de la metástasis en el cáncer colorectal. Ese trabajo es de una altísima relevancia conceptual, ha dado lugar a una publicación muy relevante en Nature sobre este mecanismo. Es una tesis que cuando lo miras, y es un trabajo que cuando lo miras con detalle parece que hay 5 o 6 tesis dentro de una, cubre muchísimos aspectos, y yo creo que es una oportunidad única tener esta presentación en directo por parte del doctor Adrià Cañellas, y espero que todo el mundo pueda luego hacer preguntas e interactuar después de esta sesión. Muchísimas gracias.
Hola, buenos días. Bueno, muchísimas gracias por la invitación a presentar. Bueno, es mi primera vez con una audiencia clínica, así que espero no decir ninguna burrada. Me cambio el inglés, que siempre lo hago así y ya lo tengo. Okay, so I've done my PhD in Eduard Batlle's lab at IRB, and in Edu's lab we are focused on studying colorectal cancer. As you all know, colorectal cancer can be diagnosed at 4 different stages. And on one hand, we find stage 4 tumors. These are patients with very poor prognosis that already debuted with metastatic disease. And for many years, we have been studying this kind of patients. But during my PhD, we decided to focus on metastatic relapse. This is the phenomenon that, as you know, happens to stage 1 to 3 patients. So these are patients that first are diagnosed with an initial primary tumor diagnosed in the colon or in the rectum, and they receive surgery and sometimes with chemotherapy, right? So these patients at this point, after surgery, they are theoretically cured. However, during the next 3 to 5 years, an important fraction of them is going to develop metastatic recurrences, mainly in the liver, also in the lungs, in the peritoneum, in bones. No? And what this clinical phenomenon implies is that before surgical resection of these primary tumors, a subset of tumor cells escaped throughout the body, and they disseminated to other organs, but they remain occult, invisible to the clinicians, no, invisible to the image diagnostics. So basically, this is what we know as residual colorectal cancer, and our knowledge on the biology of these tumors that are minuscule in nature is very poor. No. So how it's treated, this kind of disease, how we prevent metastatic relapse is much improvable. So what we decided to do was to try to understand better how are the tumor cells and their surrounding microenvironment in residual colorectal cancer. So in Edu's lab, we are mainly focused in 2 main areas. One of them is tumor heterogeneity, tumor cell plasticity and heterogeneity, and the tumor microenvironment. So regarding the first topic, tumor heterogeneity, we now know that there's a vast levels of tumor heterogeneity in a single tumor, and this is not random, but it's found to be largely reminiscent of the architecture that we find in the normal colon. No? So we know that a subset of intestinal stem cells are located at the bottom of the crypts, and they renew all the colon within a week. So basically, it's been found that despite colorectal cancers acquire mutations in key signaling pathways that regulate stemness, this hierarchy of a pool of stem cells dividing and giving rise to differentiated cells is much preserved. So basically, it's been found that in colorectal cancer, we find subsets of cancer stem cells that we can mark with LGR5. And this pool of stem cells fuels tumor growth. Basically, it's been shown that if you ablate these cells, or if you do lineage tracing experiments, this is the progeny that fuels the progression of tumors. The other topic that the lab has much interest in is the tumor microenvironment. Almost 10 years ago already, the lab made a significant discovery, which was that if you look at the genes that predict poor prognosis in primary tumors, what you find is that the vast majority of these genes are not expressed by tumor cells, but they are expressed by the tumor microenvironment, immune cells, endothelial cells, and cancer-associated fibroblasts. So after several studies, the idea that we have, our conclusion is that the tumor microenvironment is especially important to secrete TGF-beta levels. Those patients that have high levels of TGF-beta in the microenvironment are better equipped to protect themselves against adaptive immunity, and these patients have a poor prognosis. So all this data, coming back to this slide, has been demonstrated either in advanced metastatic disease or in primary tumors. But as I said before, we know very little on how this translates in residual colorectal cancer. We have basically sort of interpreted that these biological principles apply as well to residual colorectal cancer, but as there's no human samples because this is invisible disease, we don't know whether that holds as well. So we decided to investigate this, and a specific question was, given this tumor cell heterogeneity in primary tumors, is there a tumor cell phenotype that is more capable to generate metastatic disease and travel to other organs? So how we approached this question was by taking back again this list of 2,500 genes that predict poor prognosis, that predict the risk of disease recurrence. But now we focused on 99 genes that are expressed by tumor cells, by the epithelium. So as you can see, these 99 genes predict very well whether a patient is going to suffer disease relapse or not after primary tumor surgery. So now we take these 99 genes and we analyzed a single-cell RNA-seq cohort of 20 patients. No? So here we have these 20 different patients that are overlapping, or there's some patient-specific clusters, and we map the expression of this EpiHR signature. This is the poor prognosis signature of epithelial cells. And basically what we could see is that the EpiHR signature is mapping cells at different areas. So different patients have a certain amount of cells expressing high levels of the EpiHR. And what was interesting is that these cells connected to poor prognosis are not overlapping with the LGR5 signature. This is how we label cancer stem cells. So we found a subset of cells that is associated with poor prognosis that are not cancer stem cells. And as you can see, these populations are non-overlapping, and patients have a different amount of HRCs. This is how we call high relapse cells, the cells that have high levels of the EpiHR. And interestingly, those patients that are at more advanced stages, stage 3 and 4, have a higher amount of HRCs in their primary tumors. So at this point, we were obviously interested in studying what's the role of this tumor cell population to disease relapse. So for that, the lab, several years before, built an immunocompetent model to study MSS colorectal cancer So basically, what Daniele Tauriello did in the lab was to engineer mice so that they would express the four main mutations in human colorectal cancer, mutations in the Wnt pathway, in KRAS, in TGF-beta, and in p53. And he induced a recombination specifically in intestinal stem cells. So these mice develop tumors in the intestine that are less or more aggressive, and those mice that have four mutations developed aggressive tumors that invaded the adjacent areas and metastasized to liver and lungs. So now, because working with these mice was such a hurdle, what Daniele did was to generate organoids, 3D cultures of the tumors that these mice develop. So now we have a biobank of organoids that we can transplant back into immunocompetent mice. And this is how we model MSS colorectal cancer. Basically, the tumors that are generated by these, we call them MTOs, mouse tumor organoids, are reminiscent of the poor prognosis subtype of colorectal cancer with high levels of TGF-beta and CAF infiltration. Okay, so we wanted to know whether our models were suitable to study HRCs. So for that, we did single-cell RNA-seq of tumor cells that we injected in the cecum of mice. And again, what you could see is that we find a population of tumor cells with high levels of the APHR signature. And this is, again, non-overlapping with the LGR5 signature. So we find HRCs and cancer stem cells, and these are non-overlapping populations. Interestingly, we also found that those gene sets that are enriched in mouse HRCs are similar to human HRCs, and these are related to interaction with the extracellular matrix, adhesion between cells, and regulation of cell migration. And I'll be coming back to this in a few minutes. Okay, so we can study HRCs with MTOs, but it was missing this clinical aspect of the model, which is that most of these mice will die because of the primary tumor, which is something that does not happen in clinics because humans receive surgery. So what we decided to do, and this took several years, was to develop a model of primary tumor surgery with these organoids. So basically, the idea that we had was to change the injection site of these MTOs to the tip of the cecum. The cecum, I see it a bit as our appendix, in the sense that, as you can see, we generated a primary tumor, and now we can cut off the tip of the cecum with this, removing the primary tumor, and the mice are going to be fine, free of primary disease, and we can follow, thanks to luciferase that is expressed by tumor cells, the progression of the disease. So here you can see how we inject the organoids. These generate a tumor within a few weeks that we then do surgery. We remove this tumor, and the mice is going to be free of primary disease. However, over the following 3 to 4 weeks, we start seeing light reappearing, but now in the upper thorax, meaning that these mice are developing metastases in, as I said, liver and lungs. If we do this surgery very early on, we can cure these mice. We did not give time to the primary tumors to invade and metastasize. And as we do it later on, around 50 to 70% of the mice will experience disease relapse. Okay, so we were building all this, remember, to study residual colorectal cancer. So what we decided to do was to analyze livers at the very initial stages of metastasis, when we are still not seeing light, or we are seeing very few bioluminescence. So at this point, we were able to see with 3D microscopy small clusters of cells, of GFP-positive tumor cells that started to colonize the liver, and with time, these metastases progress to lesions that will end up killing the mice. So our interest was to know the phenotype, the biology of these first tumor cells that arrive to the liver and compare them to a full-blown primary tumor. But if you think about this question, it's tricky because you are interested in tens of cells in an organ that has, even in mice, over 50 million cells. So we were, I think, a bit lucky because basically, so normally you would attempt to do this with flow cytometry, no? And the protocol would be to digest the livers, no, with mincing and enzymatic digestion, and then we filter the sample to have a single cell suspension, and you would go to the fax and try to catch these rare events. But this is almost impossible, no? You are so many hours that it's not possible to profile any tumor cells. But what we discovered was that with this protocol, even though we had tumor cells initially, here you see bioluminescence, after a first filtering step, after the enzymatic digestion, most of our tumor cells were retained in the filters. So basically, with this enzymatic digestion, we digested the liver. So you can see how most of the sample flows through. However, the tumor cells are probably more difficult to digest, and they are trapped in these filters. So this simple trick, what it allowed us was to enrich over 400-fold in tumor cells. So with this trick, we were able to profile, I would say, for the first time, metastases at a very incipient stage. And what we did was, again, to do a single-cell RNA-seq experiment in which we compared primary tumors with micro, small, and macro metastases. I won't go very deep in this data, but basically, we found the usual suspects in colorectal cancer, which were cancer stem cells in green, proliferative cells, differentiated like cells that express keratin 20, and we found as well HRCs, no? Here at the top. And what really intrigued us was that the only cluster of cells that was specific of micrometastatic disease was this one in blue, and it expressed high levels of the epichr signature. Overall, what you can see is that this metastatic evolution is dynamic, and basically the tumor cell phenotypes are not constant throughout the evolution of the tumor, of metastatic relapse, but they are changing, no? Meaning that this is a plastic phenomenon, and that different tumor cell types or tumor cell phenotypes are important for different stages. So, of course, our hypothesis was that these epichr, these high relapse cells, are the first ones to arrive to a metastatic site, and then they generate metastasis. So we wanted to demonstrate this, and what we did was to look for a marker gene of the HRCs, no? And we selected EMP1, epithelial membrane protein 1, that, as you can see, overlaps nicely with the epichr signature. So in the lab, for several years, we have implemented a CRISPR technology that allows us to do gene knock-ins at specific locuses. So in the EMP1 locus, we introduced a cassette that has an inducible caspase. This will allow us to selectively kill in an inducible manner cells that express high levels of EMP1, and a TD tomato fluorescent reporter to visualize these cells. Just as a proof of concept, if we sort tumor cells with high levels of tomato, you can see how we enrich in the epichr signature, and we counter-enrich in the LGR5 signature. So we implant these organoids, these edited organoids, in the cecum of mice. And let me guide you a bit through this image. So this is the cecum of a mouse. Here we have the normal mucosa, and here the muscle layer. And we implant the organoids here. And what you can see here is that the tumor starts growing and starts invading the muscle layer. And we find that the highest level of TD tomato is found at this tumor buds or tumor cell clusters that are intermingled with the muscle layer, with these invasive cells. So these tumor cell clusters express high levels of tomato, meaning that these are HRCs, and are found in close connection to blood vessels in primary tumors. And then when we go to the metastatic sites, to the liver, we find them going out of portal veins. So basically, these data suggest that these are the cells that are leaving primary tumors and starting colonization at distant organs. Just a few things about the HRCs. Something that we found is that these tumor cells do not undergo any kind of EMT. They preserve an epithelial phenotype. Actually, we have seen how they are actually enriched in genes that mediate cell-to-cell adhesion. So we think that they undergo a collective cell migration. And we, as I said, we don't see any loss of EpCAM or ecadherin, and they do not overexpress any of the master regulators of EMT. A few other things very fast. We see that patients have different amounts or different levels of HRCs, and we have found mainly two associations. One of them is with KRAS. Patients that have KRAS mutations tend to have more HRCs. And we have actually introduced KRAS mutations in organoids, and we have seen how the epi-HR signature goes up. And the other thing is related with the tumor microenvironment. We have seen that those tumors that have more cancer-associated fibroblasts in the tumor microenvironment have more HRCs. You can see how CAFs correlate with the number of HRCs. And not only that, but in primary tumors, we find that HRCs are close to CAFs, and when we take in vitro organoids and we culture them with fibroblasts, what we see is that the number of HRCs that we have in culture is much increased. So now we are digging more into this question of why are these HRCs generated, why some patients have more or less HRCs. Okay, so we wanted to study this interchange between tumor cell phenotypes, and for that, we introduced another modification to our MTOs, now in the LGR5 locus, a marker of cancer stem cells, so that we can visualize them in green with a GFP reporter. And basically, again, what we see, this is a liver, and it's looking for a long time for very small lesions, no? A lot of cuts and trying to capture metastases at very initial stages. And what you can see is that the first groups of cells that arrive to the liver have high levels of TD tomato. But with time, this is not preserved. Let's say this is not a fixed cell state, but there's a switch in phenotypes, and other cell states, like here in green, a cancer stem cell state, are acquired. Okay, so all this data that I've been mentioning suggests now that this tumor cell phenotype that we have identified is the one that originates metastatic relapse, at least in these models. No? And for that, to demonstrate this, we made use of the inducible caspase that we had integrated in the EMP1 locus. So basically, when we give a drug that we call dimerizer that induces the coupling of the inducible caspase, and this will lead to the apoptosis of cells. So basically, the experiment that we do is we inject organoids, and after 1 week, when they have already implanted, we start ablating, giving dimerizer to ablate cells with high levels of EMP1. And we keep ablating until we do the surgery of primary tumors. Then we stop the treatment, and we will see how this disease progresses. No? So you can see how we can effectively remove cells with high levels of EMP1. And with this treatment, we do not affect the primary tumor size. So the primary tumors are able to proliferate and keep growing. However, when we monitor mice after surgery, what we see is that while control mice develop on average 10 metastases in the liver, animals that are treated have a tenfold reduction in the number of metastases, and the percentage of mice undergoing metastatic relapse is much reduced. No? So these data actually suggest that these are the cells responsible for metastatic dissemination. So we need a possibility is that by killing some tumor cells, 10–20% of tumor cells, you're inducing cell death, and this activates an immunological response. So basically what we did was to do the same experiment, but now getting rid of the LGR5 cells. And when we remove LGR5 cells, primary tumors are unaffected, but in this case, we do not see any effect in the formation of liver metastases. So this suggests that this wasn't a specific effect. So something interesting is that if we implant directly tumor cells in the liver due to this phenotypic plasticity, and we ablate these HRCs, metastases are able to form. So the HRCs are no longer important. And now the cells that mediate metastatic outgrowth are cancer stem cells, implying that these different cell states have an importance or play a role that is different depending on the time when you do this tumor cell ablation. So basically, just to recapitulate, in primary tumors, we find a subset of cells that disseminates to the body and starts metastatic colonization. However, this state is not trapped, but there's plasticity and other cell states are acquired. So with this genetic trick, we are preventing metastatic relapse, but this is still much work needs to be done to translate this into a drug. So we were interested in seeing how we can target metastases when they are at the initial stages of colonization, and this is the second part of the story, and it has to do with the use of immunotherapies. So as you all know, the use of checkpoint blockade therapy in MSS colorectal cancer is not, there's no responses, or there's very little responses, although in MSI colorectal cancers that have high numbers of mutations, you see responses. In MSS colorectal cancer, there's no responses to this kind of therapy. And what we discovered in the lab several years ago is that this type of tumors, at least in some cases, are actually recognized by the immune system. Even if they don't have so many mutations, there's still a recognition of these tumors. However, high levels of TGF-beta in the microenvironment exclude T cells to the periphery, and they are not able to infiltrate and attack tumor cells. So what Daniele demonstrated several years ago is that in purple, if you combine checkpoint blockade with TGF-beta inhibitors, you can actually get curative responses in mice. Whereas if you only give here in green checkpoint blockade, you cannot cure these metastases. So this has been trying to translate into the clinics, but as you know, the TGF-beta inhibitors have several side effects like cardiotoxicity. But at least this gives the idea that recognition is possible, but several members of the tumor microenvironment block this response. So you need to block all these pathways. So as you can see, this is a primary tumor, and we see this immune exclusion pattern, as well as we see it in the overt metastases that have grown in this model of metastatic relapse. So what was surprising to me was to see that in these very early micrometastases, you can see how there is a very high infiltration by CD3 positive T cells, meaning that these T cells, when they arrive to the target organ, they are recognized and they are heavily attacked. So you can see how the percentage of CD3 positive cells that are in metastases is actually really high at the initial stages of colonization. So in COVID time, this paper appeared by Miriam Chalabi, and well, this was COVID time. So for me, I had quite a lot of time to think, and I had this observation. And basically, I found it super interesting, no? But because what they were saying was that moving checkpoint blockade therapy, anti-PD-1 and anti-CTLA-4 in a neoadjuvant setting actually led to clinical responses, even in MSS tumors, no? These ones in green. These tumors are going to be treated with surgery anyway. So the fact that you are seeing a response doesn't mean that the patient is going to do better, no? But basically here, the cool thing would be if now you have an impact on disease recurrence. It also gives the idea that a similar type of tumor, an MSS colorectal cancer, depending on when you treat it at an advanced metastatic state or in a primary tumor, will respond differently to the therapy. So what we have done is to analyze the microenvironment of primary tumors and very early metastases and advanced metastases, no, with multiplex immunofluorescence. Basically, what we have seen, because we think that these differential responses have to do with the tumor microenvironment of these lesions, at least to some extent, is that initially metastases are heavily infiltrated with CD4 and some CD8 T cells. But over time, as they grow, there's an immune exclusion process as metastases accumulate endothelial cells and cancer-associated fibroblasts in the microenvironment. So here we order all the metastases, 130 metastases that we analyzed, and we ordered them by size. And what you can see is that very small metastases have low levels of CABs and macrophages, and are heavily infiltrated with lymphocytes. But over time, as they grow, there's a progressive immune exclusion. So, well, the idea that I had was, okay, so let's do the same experiment in our model, and let's see what happens with metastatic relapse. So what we do is we implant organoids, we implant primary tumors, we let them grow. And once metastatic dissemination, which starts around day 7 to 10, starts occurring, we will give 2 shots of neoadjuvant immunotherapy. So we do this treatment and we analyze the primary tumors. We actually see in these MSS models an increased infiltration of CD3 positive and granzyme B positive T cells. However, we did not see any curative responses in these primary tumors. However, when we monitor the mice, what we see is that the number of liver metastases is vastly reduced. Again, more than tenfold, and the percentage of mice that undergo disease relapse is much reduced. Now, meaning that neoadjuvant immunotherapy is able to eliminate the metastatic deposits that were spread throughout the body of these mice. Just as a proof of concept, if we repeat this treatment as it has been done, no, in the classic clinical trials, once metastases are overt, and many times when they have failed several lines of therapy. So if we do the primary tumor surgery, we let the metastases develop until they are overt, and then we treat with anti-PD-1 and anti-CTLA-4, we are not able to achieve any curative responses, no? On the same lines of what we observed in Daniela's paper. So this is a bit the story that we have explained. And the idea behind is that these concepts that we have seen as fixed states, like, for example, the consensus molecular subtypes that are associated with responses to immunotherapies, are actually not fixed for single patients, but there's an evolution of these of both tumor cells from a phenotypic perspective and the microenvironment, no? And this will probably have an impact on the responses of the disease at various stages. And we think that during metastatic dissemination is probably an opportunity. We know that the microenvironment is important to gain protection against immunity, for example. But during dissemination, tumor cells travel alone. So they are naked, they abandon this protective niche, and they have to recreate this protection. So there's a window of therapeutic opportunity that is not yet clear to us how long, for example, this will last in patients. Well, thank you so much for your attention, and to all the group. Thanks to Edu, my boss. It's been fantastic to work with him. And well, this is work for a lot of people, and we've also received help from external collaborators and the IRB facilities, which are fantastic. So thanks so much.